Hi everyone, welcome to the part one of the course on the concept of clinical trials. The course aims at teaching the key concept of clinical trials through the different steps of drug development, from first time in human to late stage post-marketing authorization studies. The courses focus on understanding the different type of study design and the main elements that characterize them. You will also get insight on how clinical trials are run in practice in the regulatory framework, from the target product profile to the actual submission for marketing authorization and the post-marketing surveillance. The aim of the course is not to detail the different statistical data analysis methods that might be used in clinical research, like linear models, non-parametric methods, or, for instance, Cox regression models in survival analysis, but rather addressing the specific statistical concepts that need to be dealt with, like multiplicity in terms of hypothesis testing, bias, dropouts, interim analysis, subgroup analysis, probability of success, or certain type of endpoints like biomarkers. In the first part of the course, the general context surrounding the value and use of clinical trials in drug development is being covered. Chapter 1 will focus on the definition of clinical trials and will describe the different types of trials, as well as the key features that can be applied in the design of such a trials to ensure the most robust evaluation of the research question at stake. After defining what a clinical trial is, we will see in Chapter 2 how trials are being articulated in a clinical development plan in order to generate the set of evidences needed to obtain marketing authorization granted by regulatory health authorities and ensure patient access through potential reimbursement towards the social security or insurance system. In Chapter 3, coming back to the clinical trial level, we will see what are the key steps in the planning, conduct, and reporting of clinical trials, what are the essential documents surrounding clinical trials, and finally, who are the key actors involved, namely the participants, the investigator, the study sponsor, amongst others. Finally, in the last chapter, chapter 4, we will cover the various regulation guidelines that exist to ensure that the right, safety, and well-being of trial participants are properly protected, and the result of the trials are robust and credible in answering the research question. The chapter 1 is articulated in 5 sections, covering respectively definitions and main types of clinical studies. We use here the term trial and study as a synonym in this course, which is not common everywhere. So in some geographies like Europe, the term trials will be used more specifically for certain type of studies. We'll come back on that later. Then we will illustrate with a set of examples of trials. In the third section, we will provide a short explanation on why we actually run clinical trials, to then illustrate how often clinical trials are being executed worldwide. And finally, if not obvious, provide an answer to why the use of statistics is a must in clinical trials. To start with a fundamental definition of clinical trial, we refer to the ICH definition. ICH stands for the International Conference on Harmonization. ICH brings together the regulatory authorities and pharmaceutical industry of various countries and regions in the world. Historically, the founding members have been Europe, Japan, and the US, where the objective is to harmonize the way of developing and getting to drug registration, discussing the scientific and technical requirement needed. You will hear again about ICH in the last chapter on regulation and guideline. So according to ICH, they define a clinical trial, clinical study, as an investigation in human subjects intend to discover or verify the clinical pharmacological and or other pharmacodynamic effects of an investigational product, and or to identify any adverse reactions to that investigational product, and or to study the absorption, distribution, metabolism, excretion of an investigational product with the object of ascertaining its safety and or efficacy. So the term investigation refers to the conduct of a specific experiment, a specific research with pre-specified condition. An investigational product refers to a product under evaluation for its benefits or efficacy, as well as risks or safety. When we refer to safety and risk, we refer to the adverse reaction associated to the intake of the product. When we talk about benefits or efficacy, we are looking at protecting against a disease, slowing down the progression of a disease, or curing a disease. Now, while definition refer to investigational products, in the broad, that is essentially a medicine or a drug, in a broader sense, it can be any procedure aiming at improving patient condition. And it can be, for instance, be a food regimen. It can be a cardiac device, as illustrative example. So the clinical effect refers to the benefits and or risk through the medical assessment of the patient physical condition, whether it's patient symptoms like pain, nausea, weakness, or ultimate outcome like death, laboratory examination, or other diagnostic tool like images. When we refer to pharmacological and pharmacodynamic effects, we refer to the physiological and or biochemical changes in the body induced by the administration of the investigational product. A controlled trial is a type of study in which observations made during the trial are compared to a standard called the control. What it means is that study participants are receiving either the drug and they are part of the active group, or they are receiving a placebo or an active control and they are part of the control group. The active control is typically the standard of care or the best standard of care, and you expect your drug to be better than that standard of care. Or it can be a placebo that is a substance or treatment which is designed to have no therapeutic effects. So common placebos includes inert tablets like sugar pills if it's to mimic an oral treatment, or it can be saline as inert injection if you have to compare an injection treatment. And so the control group is essential to serve as a standard or baseline for ascertaining the effect of the study drug. Indeed, what you expect is that besides the actual intervention received, whether you are in the active group or the control group, that you have a set of participants that overall are similar. And so that when you're going to compare the two groups, the comparison will allow you to really ascertain the effect of the drug. The control may be observation from a group of participants in the same trial, or observation from outside the trial. For instance, from an early trial, and that's what we call an historical control. You can easily and intuitively understand that when you have a control in the same trial, you're more likely gonna produce more robust evidence on the treatment effects than if the control is external. Indeed, if it's an historical control, there are other more sources of variability that might interfere with the assessment of the treatment effect, like different ways of assessing the patient from one experiment to another, or some of the environment differences, like different investigator doing the assessment, for instance. A randomized trial is a type of study in which participants, after assessment of eligibility and recruitment, but before receiving the intervention, are randomly allocated to receive one or the other alternative treatments. What it means is that from the set of individuals who are willing to participate in a research study, when after they have provided their consent to participate to the trial, they will be randomly allocated to one of the study groups. In this case, here in the illustration, you have two study groups, one set of patients receiving the control, being a placebo or a standard of care, and the other one receiving the investigational products. The most important advantage of proper randomization is that it minimizes allocation bias, balancing both known and unknown prognostic factors in the assignment of treatments. A prognostic factor being a characteristic of the patient that is related to the outcome, so the patient condition. As an example, if you take smoking, is a bad prognostic factor in lung cancer. And so if in the recruitment of the patient, those heavy smokers are systematically in the control group, the control group will have a less favorable outcome than those patients in the investigation group, independently of the treatment being received. And if you have this imbalance, you're likely going to bias the comparison of the two groups, and so the evaluation of the treatment effect. Randomized controlled trial is the gold standard, meaning that this is the type of design that will give you the best and the most robust estimation of a treatment effect. To illustrate a simple example of randomized clinical trial, let's consider a trial in SLE patient. SLE is an autoimmune disease in which the body's immune system mistakenly attacks healthy tissue in many parts of the body. And common symptoms include painful and swollen joint, fever, chest pain, hair loss, swollen lymph nodes, feeling tired, and red rash, which is most commonly on the face. So in this experiment, we would like to demonstrate that the percentage of responders according to the SRI-4, so that's the SLE responder index at week 24, is greater under the investigational drug compared to placebo. So to assess that objective, the primary endpoint outcome is SRI-4 response, which is assessed on each and every individual participating in the trial, and is defined as greater than or equal to 4 points. So responders is someone who has greater than or equal to 4 points reduction in the SLE disease activity index total score from baseline, so from before receiving the treatment. This SLE disease activity index 2000 is a composite endpoint that is a total score out of 24 clinical assessments of the patient. So for that this objective, the randomized controlled trial has been set up where 500 patients has been randomized into two study groups, two arms, 250 receiving the investigational drug and 250 receiving no intervention or a placebo. So the 500 patients are equally randomized in this case between the two arms. So there is a randomization ratio of 1:1. Then the patient, after receiving the treatment, are being followed for 24 weeks with regular monitoring of their SRI-4 score, but closing after 24 weeks, where the SRI-4 response rate is being compared between the two study groups, hoping that those patients receiving the investigational drug has a higher level of responders according to that SRI-4 index. So here in the example, we had a randomization ratio of 1:1. That's also part of the design of the trial about evaluating what is the best randomization ratio. And there are certain situations where it might be more adequate to have a 2:1 ratio or 3:1 ratio. As an example, when you develop a vaccine, as part of the late development, you will have, and before getting marketing authorization, you will have to demonstrate that you can produce the vaccine consistently with the same quality of the benefits and the risks. And so you will typically have a 3:1 randomization ratio. So three for one patient receiving the investigational vaccine, but from 3 different batches compared to the control. It is generally agreed that a trial with a concurrent control group in which patients are randomly assigned to receive either the test drug plus the standard of care or the standard of care alone would be the most efficient and reliable way to evaluate the safety and efficacy of the candidate products. In the context of evaluating Ebola therapies, there was some people, however, who have argued against such a design, urging instead the use of historical control, making the investigational product in the context of a pandemic as widely available as possible, arguing on ethical consideration. And then comparing the clinical outcome, in this case, the mortality, among treated patients with the rates that would have been expected in absence of the drug on the basis of the past experience with the Ebola virus disease. And so in the New England Journal of Medicine, that is a well-known peer-reviewed journal, there was an interesting debate around the advocacy on the case for the use of randomized clinical trial, arguing that the desire to allow all patients access to the investigational drug can be understood, but there are strong reasons to doubt that such a design and the use of historical control studies would allow to distinguish effective therapies from ineffective ones, and at the end might jeopardize the decision-making process in terms of marketing authorization and broader use of such a treatment in a pandemic situation. The main 3 arguments that were pushed forward for a randomized clinical trials was, on one hand, that if the 2 groups are not similar, observed different in results may be unrelated to the therapies, instead reflecting underlying difference between the groups or difference in supportive care. So without clear knowledge of the outcome, the mortality that would be expected with the study's level of supportive care, a historical control study cannot determine whether a treatment has helped or harmed the patients. As a second argument, such a trial could detect even, argument for a randomized clinical trial, that such a trial could detect even a small but meaningful benefit that an historical control, because there is more noise in the data, more factors that might add variability in the data, that an historical control trial could not credibly identify, and so miss an opportunity of a valuable treatment for the population. And the last argument was more kind of a risk management element, that when preclinical data suggests that a candidate treatment has a low likelihood of clinical efficacy, that is the previous argument, or may have a certain toxicity, RCTs, including the standard of care group, are the most efficient and reliable way to identify benefits and harm. So otherwise, it may not be possible to distinguish serious adverse drug effect from manifestation of Ebola virus disease. And so those arguments convinced about the use of RCT in evaluating Ebola therapies. More generally, the use of an external control will depend on the following four elements. Clearly, if you have an high unmet medical needs and you have a very good understanding of the mechanism of action of the molecule, and that you have a significant treatment effect, and that the endpoint is very objective robust, you can argue with the use of an external control in those circumstances. An objective endpoint, a robust endpoint, is, for instance, death, as opposed to a more subjective endpoint, less robust, like pain, where we know that each and every individual has a different assessment of pain. A synonym to non-interventional study is the term observational study that is commonly used in epidemiology. When referring to those non-interventional studies, there are two subtypes of studies. The first one is prospective non-interventional studies, and the second one is retrospective non-interventional studies. In the prospective setting, patients are being recruited and the information about the exposure is being collected at the baseline on all participants. And then the participants are being monitored until they have developed the outcome of interest. So before recruiting the patient have not the outcome of interest. The outcome of interest is monitored and observed prospectively. In a retrospective setting, both the exposure status and outcome are ascertained retrospectively. And so all the data are there, and we look backwards the information in the past. So overall, we have the two main type of studies, interventional, non-interventional. In the non-interventional, we saw the prospective and retrospective type of studies. In the interventional, we spoke about the randomized clinical trial as well as the low interventional clinical trials. And in general, the more you are on the left, the more you have a robust control. The more you are on the right, the less is being controlled. The more you are on the left, it's rather prospective and primary data collection, while more you are on the right, it is secondary use of data and retrospective. So this concept of primary data collection, secondary use of data, is another typology in the non-interventional type of studies. Primary data collection refers to a study where the data that are being collected are primarily collected for the purpose of the study. While in a secondary use of data, you are using for the purpose of the study, you are using data that already exists and that has been collected for another purpose. And this is typically with claim database that are insurance database, where you have the population with the different intervention receive outcomes, or it can be electronic medical record. And as we evolve in a world that is more and more digital, the secondary use of data in healthcare and clinical research is definitely increasing. Let's move to the example. The first example that we present here, it's a study to show the non-inferiority in terms of the immune response induced by an oral vaccine versus an intramuscular vaccine against poliomyelitis. Non-inferiority, and you will see more details in the part 2, means that the oral vaccine is as good as the intramuscular administration of the vaccine. This is an example of a randomized trial, open label, because you have the two type of administration. You have an active control, the control being the intramuscular vaccine, and it is interventional, as mentioned. There are two groups, 300 subjects being randomized 2-to-1 in this case. The randomization ratio is 2-to-1, with 200 subjects receiving the oral investigational vaccine and the 100 subjects receiving the intramuscular. And the idea of this development is just to deliver a vaccine in an oral formulation that is easier to administer than the intramuscular administration. The next example is an example of a first in man, first time in human. These are synonym study of single escalating dose of an investigational study drug to investigate the safety, tolerability, and pharmacokinetic in LT subjects. So this is typically here an example where it's randomized, it's double blind, and it's placebo controlled. So we have 50 subjects, and the 50 subjects are randomly allocated into a 1-1-1-1-1 ratio amongst the 5 groups, the placebo, and then increasing dose of the treatment. Now, in first time in human experiment, the 4 incremental doses of investigational products will generally be enrolled in a staggered manner, meaning that after 10 subjects received either 100 microgram or placebo, and that would be likely 10-2, so 10 subjects receiving 100 microgram, 2 subjects receiving placebo, and that no safety signal arises, then you would move to the next dose, with 10 subjects receiving the 200 microgram and 2 subjects receiving placebo, and so on and so forth. So really you have the incremental dose are generally staggered for safety consideration. This is a typical first time in human design for single escalating dose of the investigational drug to assess the safety, tolerability, and pharmacokinetic in a cancer patient. This typically apply what we call a 3+3 design, where 3 patients are treated per dose level. And depending on the number of dose-limiting toxicity, the dose is either escalate or the cohort expanded with 3 extra patients. Dose incremented are predetermined. So here we have the first dose, and the first none of the 3 first patients present any DLT, so the dose was escalated. And then at that dose, again, none of the 3 patients showed any DLTs during the assessment period after the dose administration, and then moved a next cohort of 3 patients to the third dose. And in that example here, we have 2 of the 3 patients not presenting any DLTs, but 1. And because there is 1 DLT, the algorithm foresee to have another set of 3 patients. If there is no DLTs, then you can move to the next dose. But when you face a situation where there are 2 patients with DLTs, it means that you have reached the maximum tolerated dose. And that will actually help you to determine the related phase 2 dose, that is the subsequent step in drug development. Here is study phase 1. That is the highest dose with a pre-specified DLT rate. So this is an old classical type of design that has been shown not to be the optimal type of design in terms of identifying in the most efficient way the related phase 2 dose. And so other approach like continual reassessment methods or the dose finding with escalation with overdose control are designed using Bayesian approach that have been proposed to have a more smooth identification of the optimal doses for the phase 2. The next example is a population-based case control study to assess the relationship between lung cancer and environmental risk factor in a certain geography here, the Appalachian Kentucky. Appalachian Kentucky has one of the highest incidence rate of lung cancer in the United States. And it was assessed that the disproportionality high incidence could not be explained by smoking consumption or tobacco consumption alone. At the same time, preliminary analysis of trace element content reveals that higher level of arsenic, notably, or chromium and nickel, in Appalachian Kentucky residents than elsewhere in Kentucky. And it was also known that trace elements could promote carcinogenesis and so induce more cancer. So these findings justified further investigation of the role of trace elements play in the development of lung cancer in this region. And so the hypothesis was that the unexpectedly high rate of lung cancer in Appalachian Kentucky was associated with exposure to environmental carcinogens that increase oxidative stress and DNA damage. And so a case control study was set up, and the case control study starts with a disease. So it starts with a patient having lung cancer in the geography, and work backward to find association between exposure and the disease. So you compare patient with and without disease to determine how they may differ in what they were exposed to. So in the case control, it's a study where the two groups are differing in outcome occurrence, so patient with lung and patient without lung. So they are first identified from the population of interest. And then samples where biological samples were taken on those subjects in order to assess the rate of moderate and high arsenic. And so look at the relationship between this rate of moderate high arsenic and the outcome of lung cancer or not. So you first have the outcome and then you look at the exposure. So of note, the study is considered as non-interventional when the biological specimens are collected as part of standard practice, but would be interventional if these samples would be specifically collected for evaluation of the study objectives. This is another non-interventional study example. It's a population-based case control study again of lung cancer and green tea consumption amongst women living in the Shanghai in China. It has been a retrospective study. So there was epidemiological evidence regarding the association between the consumption of green tea and lung cancer. But those evidence were limited and inconclusive. Although some experimental studies showed consistently that tea preparation and tea polyphenols may inhibit the induction of a variety of cancer, including lung cancer. And so in this population-based case control study, the author examined the association between past consumption of green tea and the risk of lung cancer. So they identified about 650, 649 incident case of primary lung cancer amongst women diagnosed from February 92 through January 94 using the cancer registry in Shanghai. And then they randomly selected a control group of 675 women from the same geography, but from the residential registry. And they matched according to age distribution of the case. And then green tea consumption was ascertained through face-to-face interviews, but looking retrospectively. So the authors estimate the adjusted odds ratio and 95 constant interval using unconditional logistic regression. Among the non-smoking women, consumption of green tea was associated with a reduced risk of lung cancer with an odds ratio of 0.69, and the risks decreased with increasing consumption. However, they found little association among women who smoked. And essentially, when looking at the past literature, the author conclude that the inconsistency in the association between drinking tea and the risk of lung cancer reported in previous studies may be in part be due to the inadequate control of this confounding of active smoking in the design and the analysis of the results. As a last example, we consider a burden of disease cervical cancer study in Tanzania. There is also a non-interventional type of studies Disease burden is looking at the impact of the disease in terms of health problem on an area measured by financial cost, mortality, morbidity, or other indicator. And so it helps to factualize the unmet medical needs associated to the disease. And so here it's a cohort of 105 female aged between 30 and 75 years in Tanzania with cervical cancer, stratified according to the severity of the severity of the cancer, so the stage of the cancer, and looking at measuring a primary outcome in terms of socioeconomic burden to the household, as well as some secondary outcome measure relates to the projection of economic impact on the community. Why do we perform clinical trials? The answer is fairly straightforward, and we can refer to the Aristotle philosopher in the Metaphysics, where Aristotle just had the quote about all men naturally desire knowledge. So clinical trials and clinical studies are being conducted to advance in medical practice based on research, and that the research validity is based on the scientific credibility of the experiment being conducted. And so, bottom line, we perform clinical trials to answer a question in a scientific manner through performing valid research. And that the scientific manner goes through the scientific method that is starting from a question and designing and planning and designing an experiment, conducting the experiment with the end goal in mind, that is the question, answering the question, to then collect the data, analyze, and conclude. And that's typically the proper scientific methods of a quality by design approach, starting with a clear question to answer so that the way of designing the experiment is optimized to answer those questions in a robust manner. How often do we perform a clinical trial? Interesting question, as it's quite significant, the amount of clinical research being conducted worldwide. There's an interesting website named clinicaltrial.gov that was created in order to promote transparency and sharing of study results across the different sponsors of studies, whether academia or pharmaceutical industry. And in 2004, it was effective in 2005, the International Committee of Medical Journal Editors required that all clinical trials be prospectively registered into this website, clinicaltrial.gov, and that at the end of the trial, some key results is being disclosed. And that has been a push from the medical journal editors to promote transparency and sharing of information to avoid the risk of certain sponsors, whether academia or pharmaceutical industry, not disclosing result of certain type of studies, and notably those studies that failed to demonstrate the benefit of a certain treatment. And so really the intent was to share as much as possible to optimize research and avoid that we repeat the same mistakes. So when looking into the clinicaltrial.gov, what we see is that there is about 400,000 clinical trials registered starting in 2000, but with an inflection point in 2005 with the ICMGE decision, and then supported by the Food and Drug Administration Amendment Act in 2007, who is also expanding the expectation in terms of registration requirements from FDA, Food and Drug Administration standpoint, FDA being the US health authorities evaluating the marketing authorization of a new drug. I was mentioning that those trials are from the various sponsors, whether they are pharmaceutical companies or non-commercial sponsors like academia. In the European economic area, approximately 60% of the trials are sponsored by the pharmaceutical industry and 40% by non-commercial sponsors, mainly academia. Ronald Fisher is known for his impressive contributions to statistics that he notably developed during his work at Rothamsted Experimental Station in the UK, with the main two contributions around the principle of design of experiments and the analysis of variance. Although developed in the agriculture research, most of these principles are fully applicable to medical research. He actually summarized well the importance of statistics through his well-known quotes. To call in the statistician after the experiment is done may be no more than asking him to perform a post-mortem examination. He may be able to say what the experiment died of. And this shows that statistics is needed at every step, from the design, from the planning of the experiment, thinking about what is the appropriate control, what is the right way of randomization. Do we need an internal control or would an external control be appropriate? How do you define the endpoint? How are you going to answer the question through the statistical analysis? Is really across all those different steps. And so, again, this shows the importance of the scientific methods with a quality by design approach, starting from clear question and then prospectively planning the experiment as opposed to running an experiment vaguely and then not controlling for different factors that might induce noise into the data and confound, and then realize that you cannot address your research question. Another illustration is the example of this experiment that was designed to assess the efficacy and safety of a novel formulation, the FX06, in patients with osteoarthritis of the knee. The experiment was designed to compare three treatments. So patients were randomized according to the three groups. The first group is the control, the initial existing formulation with 40 microgram, compared to two novel formulations for sustained release, one at 40 microgram and the other one at 10 microgram. What you see on the x-axis is weeks post-treatment, while on the y-axis you have the least square mean change from baseline, that is an average daily pain intensity change from baseline. What you see is if you only observe the first 2 weeks, there is no much discrimination between the different treatment arm, and it's only later between week 2 and week 3 that the respective treatments discriminate, with clearly the original formulation getting quickly back to normal, meaning that there is no much effect on pain, while the new formulation have a more sustained benefit. And you can also see the the dose effects between the 10 microgram and the 40 microgram, where you have an even further stronger effect at 40 microgram. But the sustainability and the duration is very similar between the 10 and the 40 microgram. So what this illustrates, again, in terms of the design of the experiment, is how long you're going to assess the patient and monitor the patient, and how you're going to analyze the longitudinal data in a way that you can dissociate discriminate the treatment during the course, over time properly. Another example is an experiment where the primary objective is to demonstrate that the active treatment improve the allergic symptoms compared to placebo. So the primary endpoint outcome is the discomfort score. That is a combination of different symptoms scores. So it's a total score related to sneezing, itching, and wheezing. And what we observe in this experiment, again, we have on the x-axis time, we have the disconfirmed score as on the y-axis. And while the placebo is a substance not containing active ingredient, there might be situations where receiving this fake treatment will improve the patient condition because the participant has the expectation that it will be helpful and will improve the condition. So the more a person believes they are going to benefit from the treatment, the more likely is that they will experience a benefit, whether they have an active drug or not. And this phenomenon is known as placebo effect. It's very well known in studies involving painkiller or anti-allergic drugs. So if one limits the observation to the first few weeks, the experiment conclude on no added value of the active treatment, while beyond 3-4 weeks of assessment, it's obvious the active treatment improved the patient condition. Related to this, there is an interesting publication in 2011 that look at placebo effect, and where placebo effect was the highest. And the author notably observed that the placebo effect was the highest in allergic rhinitis, so similar to the example I shared before, as well as in asthma, but less in allergic skin. They also observed that the placebo effect was significantly higher in studies in which the clinical outcome or measure is subjective, compared to when it's an objective indicator and an objective endpoint, which relates to some of the element I shared before on the robustness of the endpoint. So statistics is an important tool to ensure the scientific validity of the experiment and the robustness of the conclusion in a cost-effective way. Statistics contributes at 4 levels. One is towards an adequate study design to collect the appropriate data for answering the specific question of interest based on the study rationale. So being clear on the target population, inclusion, exclusion criteria, select the appropriate control group, think about the different treatments that are relevant to compare based on the scientific question, and the most adequate sampling methods, randomization, stratification, that you will have more detail in part 2, as well as the blinding, for instance. The second point is the proper sample size computation. so that there is proper risk management. If you design a trial with too much patient, with too many patients, you will have more information than needed. And so you will expose more individual than needed, plus it is costly. If you have too few participants, you have the risk of not being able to conclude because of the variability. And so the proper sample size computation is needed to ensure that the study is adequately powered and that depending on the type of objective, you have to assess that you allow a proper control of type 1 error to evaluate the study objectives. So here it's important on whether you have one primary objective or you have more than one primary objective and need to handle the multiplicity. Again, this will be addressed in details in the part 2 of the course. And the third element is the proper approach to control and minimize bias. We spoke about randomization and blinding that are in place to control and minimize potential bias. And there is a bias when the observed difference between study group is due to another factor than the treatment under study. And so there is confounding. Finally, the broad range of statistical methodologies that permits to transform the data collected into useful evidences, information, ranging from descriptive summaries, tables to inferential comparison between groups are key consideration as well, so that the appropriate statistical methods is being applied. Let's take example illustrating a different source of bias in clinical trial. So a bias is any process which tends to produce results or conclusions that differ systematically from the truth. A first example is the evaluation bias, when the measure of the endpoint is not performed the same way in the different groups. So one example would be that patient in group 1 would be assessed by one clinician, and patient in the other group would be assessed by another clinician. And so there is the risk that the way the measure is being performed on the endpoint is different because of two different assessors. In general, randomization and blinding of the assessor should limit this bias. Another source of bias is called analytical bias that arises when results are evaluated with a non-adequate methodology, giving a bias estimator. The bias of an estimator is the difference between an estimator's expectation and the true value of the parameters being estimated. As a very simple example, you can think about the estimator of the variance when you use either, you divide either by n or n minus 1, the sum of square. This analytical bias is not to be confounded with reporting bias that involve a skew in the availability of data, such that observation of a certain kind may be more likely to be reported and consequently used in research. Reporting bias might be triggered, for instance, in an open label study when either the investigational product or the control has a known potential safety risk. Suppose it's with the investigational product, and knowing the potential risk, the participant and treating physician, the assessor, will likely pay more attention to any occurrence of such or similar event if the patient is receiving the investigational product, so randomized in the treatment arm with this potential safety risk, while not the case if the participant is in the other group. Another source of bias is confusion bias that is actually an example of analytical bias as well. It arises when other factors called confounding factors are not taken into account, while they may induce variability in the results and impact conclusions. Such factors should be proactively controlled via adequate study design and statistical methodology. So if we look at this example where we have two drugs, drug A and drug B, and the outcome is success or failure. And what it appears is that treatment B has a higher rate of success compared to drug A, a patient receiving drug A. Now, looking again at the difference in success proportion between drugs, but separately for each baseline biomarker status, as negative as positive, it is less obvious whether drug B is better than drug A on the contrary. So we have 100% for drug A, 94% drug B, 57%, 50%. Note that these results need to be considered carefully because the ratio of patients who receive drug A and drug B is quite different between the two biomarker status. So you have a ratio 1 to 3 here, and you have a ratio 7 to 1 here. As we will see later in the course, it is important to identify such prognostic factors of the biomarker status in designing the study to either restrict the target population through the protocol inclusion-exclusion criteria, or to stratify the recruitment of patients in order to have treatment allocation equally distributed in both biomarker statuses subpopulation. In addition, in the current example, there is very limited patient with positive status of the baseline biomarker who received drug B, 10 patients. And it might not be appropriate to consider an asymptotic confidence interval, but rather use an exact confidence interval or a Fisher-exact test. If you would apply a Fisher-exact test, you would see that the difference is not significant. On the other hand, it definitely questions the overall analysis because this lack of significance might be linked to the sample size, but in all circumstances, it questions the overall analysis indicating that drug B is better than drug A. So this simple example illustrates how a confounding factor, in this case the baseline biomarker status, might impact conclusion or at least impact the ability to conclude if this is not properly taken into account even from the beginning. Now obviously through randomization, for instance, you could expect to minimize the risk of such an imbalance, but still using stratification as it will be described in the part 2 is very valuable when the prognostic factor is of significant effect. So how to generate valid data in clinical trials? We have two tools. One is randomization, which ensure minimization of bias at study entry, and blinding, which ensure minimization of bias during the clinical trial with regard to treatment effect. As explained, randomization balances both observed and unobserved characteristics between the study groups, so it accounts for all possible confounding variables. In the situation where randomization is not possible and the use of external control would be considered as adequate to generate robust enough evidences for decision making, it's important to take the appropriate methodology, because even after applying stringent inclusion-exclusion criteria between your investigational trial and the external control, the distribution of patient characteristics may be different between the trial arm and the external control. So statistical approach should be thoughtfully considered to further increase population comparability and minimize bias. And that's where propensity score methodology is commonly used, historically more in epidemiology, to achieve greater balance in patient distribution. So a propensity score is the conditional probability that a subject receives treatment given the subject observed covariates. And the goal of the propensity scoring is to mimic what happens in randomized controlled trial by balancing observed covariates between subjects in control and treatment study groups. And so propensity score matching refers to the pairing of treatments and control subjects with similar value of the propensity score. So you're kind of creating blocks of treated patient and control subjects who are as likely as possible towards this conditional probability of receiving treatment or control based on those observed covariates. And that helps in the analysis to take into account about this pairing and matching in the analysis so that the comparison is done within those subgroup of matched patient. And you will see in the part 2 on randomization the concept of randomized block design, and it's a bit of the same idea here with the blocks. So this ends the chapter 1 about clinical trial with the following key learnings. First, randomization and blinding are two essential instruments to design robust clinical studies. Second, there are various types of study design, and notably studies ranging from being interventional or non-interventional, with internal or external controls, being randomized or not, with various levels of blinding. And finally, thirdly, statistical expertise is essential to answer a question in a scientific manner, really from the planning, from the design perspective, to the analysis and actual reporting of the result and interpretation of the result. You are now invited to switch to the next video that is related to the chapter 2 on clinical development.